Couple then. Hello, good evening, people. How are you? Komoda, can you hear me clearly? Audible, ne? I'm audible, right? So, Komoda, what are you doing? Did you you had school today, ne? You went to school today, ne? So, good evening. Right. So, people, today we'll have a short discussion on proteins and nucleic acids. Proteins and nucleic acid short discussion, right? So, we'll go like this. The old sections will revise again and again, right? We'll go like that with the previous sections. So, I thought of discussing with you today the those two proteins and nucleic acids. Proteins and nucleic acids. Proteins and nucleic acids. Right. Proteins. Last time we discussed carbohydrate. Ne? Did we discuss lipids? Yes or no? Did we discuss lipids? Yes or no? We didn't do lipids. Right. Lipids. I think we have to start from then lipids. Ne? So, we will quickly go through lipids. Carbohydrates we discussed in the previous class. Carbohydrates we discussed in the previous class. Shall we discuss quickly lipids and then we will move into carbohydrates? Lipids. So, what do you know about lipids? Carbon, hydrogen, oxygen containing compounds. Ne? But we learn people they are not macromolecules. They are not macromolecules. They are not macromolecules. And then they are not polymers neither. They are not polymers also, right? And then lipids. Lipids. So people, lipids. Lipids. Long time ago, ne? We did that once upon a time, long time ago. Still, I had to go down. Right, people, we learn that lipids are quite heat stable. We learn oxygen content is very less. No hydrogen oxygen to ratio is not 2:1, right? Hydrogen oxygen ratio is not 2:1. Biologically important types of lipids, we said fats, phospholipids and steroids. And the fats, phospholipids and steroids are biologically important lipids, right? Biologically important lipid types are there. And then people, fats, We started our discussion with fats. Fats are made up of glycerol and fatty acids. Glycerol and fatty acids form fats. Glycerol, you know, ne? Glycerol, you know, a molecule with 3 carbon like this. Glycerol is a molecule with 3 carbon and 3 oxygen. OH, ne? 3 OH like this. 3 OH. This is glycerol. Glycerol, alcohol, right? And then we learn people glycerol reacts with fatty acids. Fatty acids we wrote in a simple way like this. We wrote it like that. But you learn that people this R represent a hydrocarbon chain. So it's a long carbon hydrogen containing part R. So if you like, I can write it like this. Carbon, 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 carbon. How many carbon you write in this chain? How many carbon you have to show? You have to show people 16 to 18 carbon. Neither. Please chat with me, right? Otherwise, I'm simply talking with the camera. Neither. Right? I don't like that. I think I have some live, bright and live students. Ne? I think so. Yes, yes, sir. Okay, someone is chatting with me. Someone chats with me, so let me, okay. Santosh, okay. So, this reaction happened 3 times. We said that the reaction goes like this. This is removing water molecule. When water removes in the reaction, what do you call the process, people? What do you call the process? Can someone tell me what you call the process? What you call process? Condensation, eh? Condensation reaction, right? Condensation good. That is myelin, eh? Myelin. I think I'm correct with the pronunciation. Myelin, right? So the condensation reaction happens. So this condensation reaction happened 3 times. And then, so it's formed, once it happened, it's a monoglyceride. It is a monoglyceride. So, you know, this condensation is going to introduce a new bond. That bond is the ester bond. It goes like that. So, the process can repeat 2 more times. So, when it happen again, the resultant molecule is a diglyceride. When it happen once again, the resultant molecule is a triglyceride. The triglyceride. So how many water molecules are going to remove during the process? 3 water molecules are going to remove during this process, right? So that's what you learn. And the molecule is a triglyceride. You learn, people, this triglyceride we can simply show. We can simply show the triglyceride. We can simply show like this. We can show the glycerol part, and these are the 3 OH fatty acid tails, fatty acid chains like that. So we can show the triglyceride like this. But you learn when there is a double bond present in the chain, when the double bond present, the tail bends like that. When a double bond present in the fatty acid, then what do you call that, people? Unsaturated fatty acid. So sometimes some triglycerides are unsaturated, right? And then you learn, people, 2 types of unsaturation. Who can remember that? There are 2 types of unsaturation. Very good. Santosh says cis and trans. And Tabhana, right? Jai Prakash, right? And then trans and cis and trans, that's correct, ne? Cis and trans, that's good, very correct. So if fatty acids are, what do you call this one, of course? What is this one, people? This one, hydrogen present on the same side. When hydrogen present on the same side, what do you call that? Cis, ne? It is cis. Cis unsaturated ones, They don't pack closely. So therefore you learn that these triglycerides are liquids at room temperature. And when they are trans, so let me draw a trans one. This is trans 1 people. Trans 1, the tail is nearly straight, right? Tail nearly straight, does not bend much. So trans means they are present on opposite side, the hydrogen on the opposite side. Tail is not going to change. Tail is still nearly going to be straight. So therefore, these are trans-unsaturated. Trans-unsaturated ones can pack closely. Therefore, you learn that they are solids at room temperature. The cis ones are liquid at room temperature. Liquid ones we call them as oils. Solid ones we call them as, what do you call them? You call them as fat. Solid ones you call them as fat. So check here people. Fatty acid molecules bind to each hydroxyl group of glycerol. Hydroxyl group of glycerol. Hydroxyl group of glycerol. Oops. Hydroxyl group of glycerol. And then fats are made up of glycerol and fatty acid, right? And then fatty acid molecules bind to each hydroxyl group by forming an ester bond. Ester bond. So you can see that saturated fat made up of saturated fatty acid, unsaturated fat made up of unsaturated fatty acids with hydrocarbon chains having one or more double bonds. Usually plant fats come under this category. And then people, butter. Look at this. Butter. What is that? This is butter is saturated fat. And then we have unsaturated fat. Usually plant fat comes under this category. Plant fat come under this category. Plant fats come under that category. Plant fats can different oils and all. Coconut oil, sesame oil, vegetable oil, right? Mostly liquid in room temperature. Example, vegetable oil. Unsaturated fats are classified based on the nature of their double bonds. Cis unsaturated and trans unsaturated. And consumption of excess saturated fat and trans unsaturated fat. This is important point. What is that? Consumption of excess saturated fat and trans unsaturated fat, right? Contribute for atherosclerosis. Remember what is atherosclerosis? What is atherosclerosis, people? What is that condition? What do you call as atherosclerosis? Sclerosis is hardening. Sclerosis is hardening. Athero means blood vessels, hardening of the blood vessels. So what happened? The blood pressure rises because blood vessels must be elastic. The blood vessels now become harder. So that condition we call as the atherosclerosis, right? Sclerosis, atherosclerosis. So that's bad. What fat is not good for your health? You can take people trans unsaturated and saturated fats are not good. Unsaturated fat, cis unsaturated fat is healthier as they mean, right? As they have mentioned. Now people, thereafter we learn about the phospholipids. Lipids containing phosphate is phospholipids, right? You learn people, it's like the same arrangement. It is the same arrangement but with a slight modification. What is that modification, people? One of this position has a phosphate, right? It's a triglyceride, right? But similar to a triglyceride. But here what do you have, people? Here there is a phosphate part. There is a phosphate here. So you call it as phospholipids. Phospholipids. Now phospholipids are special because these phospholipids, they have glycerol. This is glycerol part. I'll show you the long hydrocarbon chain like this. So this is the fatty acid part. This is the second fatty acid, right? This is the second fatty acid. Third position is a phosphate, ne? Phosphate is negatively charged because of this phosphate part. And you can see here, this entire part is polar. This entire part is polar. This entire part is polar. What do you call polar? Polar head. We call it as polar head. And then what do you call these two people? These two, what do you call? Non-polar tails. Non-polar tails. So how many non-polar tails present? Two non-polar tails present. So phospholipids, you can see head and tail structure present. Phospholipids are having head and tail structure. You learn sometimes some special charged groups are attached to the phosphate part. Can someone tell me the name of it? Name of it, choline. Very good. Very good, very good. Right. So you're doing well, ne? That's choline. You don't have to know the arrangement of choline, but anyway, it's attached here. Choline, ne? Choline. And then this choline is a charged part. So choline together with the phosphate, together with the glycerol, together with these carbonyl parts, everything forms the polar head. So these phospholipids we can draw in a different way. Phospholipids we can draw in a different way. Neither head and tail structure we can represent in a different way. Look at this. Look at this. We can show it like this. We can show it like this. This is the polar head and the two non-polar tails. Head and tail structure. You learn this arrangement in lipid. This arrangement of phospholipids you learn in cell membrane lesson. Cell membrane has the key material forming the cell membrane is the phospholipid bilayer. Phospholipids, they form a bilayer like this, and this phospholipid bilayer forms the plasma membrane and other membranes like the nuclear envelope, mitochondrial membrane, chloroplast membrane, Golgi membrane, ER membrane, lysosome membrane. They all have this bilayer arrangement. Bilayer arrangement. You learn, people, this part is called inward. The inward, heads are turned towards outward. Heads turn towards outward. Head turn towards outward. So heads are polar. So they like to interact with water present outside and inside of the cell. You learn all these details. You learn all these details. Right, people? So let's have a look here. Phospholipids. Right, time to read little bit. Right, time to read. Phospholipids are major components of the cell membrane. They are composed of two fatty acids and one phosphate group attached to one glycerol molecule. The phosphate groups gives the negative electrical charge to phospholipid molecules. Typically, an additional polar molecule or small charge molecule also linked to the phosphate group. The two ends of the phospholipid shows different behavior, and we call this as the amphipathic property. Amphipathic people, amphipathic, amphipathic, property, right? Polar, non-polar property present. That's not mentioned here neither, right? Anyway, amphipathic property is there. So you can see the choline and phosphate and glycerol part. So here the lipids. So you don't have to learn about cholesterol. no need to learn about cholesterols, right? So functions of lipids. Can you tell me functions of lipids, what they do inside your body? Involving as a food reserve, neither, and maintain fluidity of the plasma membrane, act as signaling molecule, because you know some are actually hormones found as component of the cell membrane. So those are functions of the lipids, right? Functions of lipids, right, people? So that's all about lipids. Time to discuss about proteins. Time to discuss about proteins. So many questions I have. Mariam, transcends is okay. Thank you, Mariam. Choline, Hari. Rishma says choline, Hari. Methulimane says choline, good. Transcends is nice. Now, proteins. Now, you know proteins, people. Proteins are proteus, the main component. So, what's the main material present in your body other than water? What is the most abundant material in your body? We take it as protein. Proteus. In plant body, you can see that the main material is carbohydrate, because cellulose, hemicellulose, lignin, starch, whatever, pectin and all these are carbohydrates. And then in human body, we don't have cellulose, right? You're not a tree, so therefore you have protein as the main material other than water. Next most abundant material you can take as protein if you take as a compound. And proteins, we can see, people, made up of carbon, hydrogen, oxygen, nitrogen and sulfur. So these proteins are polymers of amino acids, amino acid polymers, polymers of amino acids, proteins. And then, people, in an amino acid, you learn the structure of an amino acid. Amino acid structure you learn. Central carbon atom present, 4 groups are attached like this. This is COOH part, carboxylic acid group. This is hydrogen part, and this is variable R group. And then this is NH2 amine group, or amino group, is available. 4 groups are attached to the central carbon atom. You call this as alpha carbon. It is alpha because it's a numbering system. This is the first carbon, and thereafter the other carbon, alpha, beta, like that you count. So this is alpha carbon. Carbon present next to carboxylic group is the alpha carbon. And then you learn, people, the backbone. Remember, this part you call as the backbone. This part is the backbone. And then this part R is variable. It's variable so that there are 20 different types of amino acids present, right? And then you learn, people, this R is forming side chains. So amino acids are forming a long chain. They react. You learn also, people, the way that the two amino acids react, way the amino acids react with one another, right? The way that the amino acids react. H2N, CH, R, C, double O, OH reacts with another amino acid, like this. So this reaction you learn as a condensation reaction. Here you can see, people, here you can see the water molecule is going to be released. So condensation reaction. So you get this. So it results what, people? Peptide bond. It results a peptide bond. So this is the resulted dipeptide. So you can see this new bond is the peptide bond, and then the resulting molecule you can call as a dipeptide, right? Resulting molecule is a dipeptide. You find it easier now, you find it easier, when you learn this for the first time, people, this lesson was little complicated. Now you find it easier, I'm sure you find this easier. So you got to be familiar. That's how you learn. You read it multiple times. Those information become more familiar with you, and thereafter people it's not a strange lesson. And then when you read the same note 5 times to 7 times people, then you remember it fully, right, forever. That's simple learning. You have to come into this track, right? So this is the peptide bond, right? If you are not lazy and if you become a frequent reader, these notes, you read it frequently. When you forget, you read. When you forget, you read. And when you do that, you remember this information better. And then when you remember it better, then the things become interesting, right? Things become interesting when you remember it. And so that's really useful, right? Things become interested, you can learn something new, and then you are ready to learn something new, right? So that the learning process then starts, right? You learn, you read, you remember, you can learn something new. How about in the other way? You learn, you don't remember, you don't understand, you don't find that interesting, then it become boring, and then you can't learn something new because you don't know the relation, right? You're not ready to learn something new. Okay, so that's the learning process. So participate in this night session, right? We are going back in time. We do a time travel simply here. So go back and then learn some early lessons once again. This will really help you because I'm giving you a small push, right, for you to right, revise the sections. So the peptide bond is going to form, and this is a condensation reaction. You learn people water. You learn that water is going to release from this reaction. You can't see that neither, but anyway water removes from this process. Dipeptide form, and if you understand the way of forming dipeptide, polypeptides are going to form on the same manner. Only thing is this is going to repeat. This is going to repeat multiple times. This is going to repeat multiple times. So we have, then we have the a long chain. So look at that, people. When the process repeat hundreds of times, we learn 100 to 300 times this can repeat, joining so many amino acid. So that's a protein. That is a protein. And then each circle is an amino acid. So I'm showing you people these variable R groups. They are different to one another. Short and long variable R groups are there neither. So they form the side chain. So we have the main chain and side chains are there. So this is the first level of the protein structure. What do you call that, people? What do you call this level of protein structure? What do you call it? What do you call this? Primary structure. Very good. I drew a cartoon in the class, right? Primary structure is like when you are naked, and when you dress a little bit, you become secondary. You dress neatly and put the tie and wear shoes, people, then it becomes the tertiary level structure. You find someone similar to you, neatly dressed one, then you together makes a new protein. What do you call that? Quaternary level structure, right, people? So primary level structure is the most diverse, because you have to understand there are 20 different amino acid at each point. So this one can be one of those 20. This can be one of those 20. So therefore primary structure has a very high diversity, right? You learn that the different types of proteins are there depending on the variation. Each amino acid has 20 options. Each amino acid has 20 options. So we all animals, we have same amino acids, but this order is different. So when you eat meat, beef, when you eat beef or when you eat prawns, you get 20 amino acids. But in prawns and in beef, those amino acids are in different order. But it's the same amino acids, right? And we break them down, we absorb the amino acids, and we join these amino acids, people, according to our information, our DNA given information, so that we make human protein. Ne? So what makes a human protein different to a cow protein? That is the primary structure difference. In other words, people, the primary structure is the order of amino acid. Primary structure is the order of amino acids. Right? Primary structure is the order of amino acid. Right? So primary structure. So we learn, people, this primary structure, the distant amino acids, they are forming attractions like this. Distant amino acids are forming attractions. As a result, the primary structure is going to fold. Neither. As a result, the primary structure is going to fold. Let's take that these distant amino acids are now forming attractions with each other. This is going to coil the primary structure, and as a result we obtain secondary structure. So what makes a secondary structure, people? The hydrogen bonds. And you learn, people, these hydrogen bonds are present between the hydrogen of the amine group and oxygen of the carboxyl group. So here this is the hydrogen attached to nitrogen, and then here oxygen attached to carbon, carboxyl group. Between them, this is one amino acid, nitrogen, attached hydrogen. This is a distant amino acid, carbon, oxygen attached to carbon. They form the hydrogen bonds. As a result, the chain is going to fold, right, frequently, and then forming, people, two structures. Can someone tell me those two structures, secondary structures and their names? Can you tell me the secondary structures and their names? Alpha helix and beta pleated sheet. So alpha helix is a helical twisting like this. Beta pleated sheet, on the other hand, is a zigzag way folding. Right? So both stages we consider as, people, secondary level structure. So this is going to thicken the protein. The thickness of the protein fiber increases, and on the other hand, people, we can see that the length is going to reduce. So that the secondary level structure. So let's see your note a little bit, people. Let's check the note a little bit. Right? So we discussed this part. Amino acids, how the condensation take place, and then levels of protein structure. Primary, the unique sequence of linearly arranged, amino acids linked by peptide bond is the primary structure of the protein. Right? Secondary structure, read that part, people. The secondary structure, the primary structure of the single polypeptide chain coil and fold and result intramolecular hydrogen bonds between oxygen atoms of the carboxyl group, and the hydrogen atom of the amino groups of the same polypeptide chain. So there are two arrangements here. Alpha helix, example is keratin, beta pleated sheet, spider silk fiber. Beta pleated sheet, spider silk fiber. So you can see these two structures, alpha helix and the beta pleated sheet. So moving to next part, right, tertiary level structure. It doesn't matter, you did this in school also. So you are learning couple of times the same thing, but it's an advantage because you have to read, as I said, 5 to 8 times the resource book, the same content you have to go again and again and again. So we did it in the class one time, you did it in the school second time, you studied it third time, in the BioEve class fourth time. So a couple of times you have to now go through the section. So do them, right? This will help you to memorize more and more. Don't take this exam easy because there are so many things you have to memorize, right? You will feel overwhelming after some time. right? Because so many information flow, biophysics, chemistry, ne? Right? And you will feel the weight of it soon. And so take it cool, ne? Take it cool. Just follow our instructions, work with us, and you will find that these things are really right, not serious, and you can learn them effectively, right? Tertiary structure usually secondary polypeptide chains bend and fold extensively, forming precise, compact, unique, functional and three-dimensional shape, resulting from interaction between side chain R groups. Right? Look at these people. Tertiary level structure you learn. Tertiary level structure you learn. The protein molecule become ball-like. It become a ball like structure. So we said that this secondary level structure, it further fold. I use this diagram in the class, and then people, these folding is supported by sulfur containing bonds. Supported by sulfur containing bonds. We call them as disulfide bridges. So disulfide bridges are helpful to fold the chain. But you know, you understood people, not only disulfide bonds play a key role here in formation of the tertiary level structure. What other bonds are helping in forming tertiary level structure, my dear? What other bonds? Ionic bonds are involving, very good. And then hydrophobic interactions are involving, very good. And then what else? Hydrogen bonds, hydrophobic. Is that all? Van der Waals interactions, that's correct. Right? So these are the different interactions. Right? Van der Waals. How do you write Van der Waals? VANDERWAALS. Van der Waal. So we can see hydrophobic, hydrophilic R groups are going to turn outside. These are hydrophilic R groups, hydrophilic, water-loving ones. They turn outside, so that this level, the ball-like protein is water-soluble. I'm showing you these are hydrophobic R groups, triangular shaped ones. I'm taking them as hydrophobic R groups. Hydrophobic R groups are turning inside and then attracting each other. In addition to that, what are these people? Hydrogen bonds. In addition to that, these are the ionic bonds and van der Waal interactions. All these things are helping to maintain this what do you call ball-like arrangement, this tertiary level structure of a protein. Tertiary level structure of a protein. You have to understand, all proteins don't undergo all these 4 different structural levels, right? All people don't marry, some people, all people are not even nicely dressed, some are street beggars are there, they don't wear the full suit, you can dress for a party or some special function, you dress like that, but everybody don't dress like that, so you can't expect the proteins to build up all 4 levels for every protein. Some proteins are having only primary, some proteins are having primary and secondary, some proteins are having primary, secondary and also tertiary. Some proteins are go beyond that primary, secondary, tertiary and quaternary level structure is there, right? So this is a tertiary level protein. Can you tell me one example of a tertiary level protein? Who's going to tell me? Very good, Santosh says myoglobin, right? Santosh means what? Happy, Santosh, your name meaning is happy, right? Yes, that's good, Santosham, right? So myoglobin, albumin, that's correct, very good, right? So most of the enzymes, that's also correct. Jayaprakash says that most of the enzymes, that's correct. Yeah, most of the enzymes, because some enzymes are RNA molecules, right? Some enzymes are RNA molecules, so we can't take that all enzymes. right? Anyway, tertiary level structure is this. And then people, we go for the next level. What is that quaternary level structure? Two tertiary can come and join and then build up the quaternary level structure. And then again you can see that the weak interactions are involving in forming the quaternary level structure. So you learn here, usually secondary polypeptide chains bend and folds extensively and then forms the quaternary level, sorry, tertiary level structure, and then hydrogen bonds, oops, these hydrogen bonds, disulfide bonds, ionic bonds, hydrophobic interactions, van der Waals interactions. So they are examples. Then people come to the quaternary level structure. Aggregations of two or more polypeptide chains are forming the quaternary level structure. Separate chains are called as protein subunits. That's important, eh? Protein subunits. Dear, protein subunits. Protein subunits. So how many subunits present in hemoglobin? How many subunits present in hemoglobin? You can see that nice diagram, beta subunit and alpha subunit. How many 4 subunits are there in hemoglobin? What is the red color part, people? What's the red color part? You have to understand that hemoglobin is a protein, gas transport protein in your blood. Neither. Red color part is the heme part, right? Heme part, so that helps in oxygen transport, right? Hemoglobin. Denaturation of proteins, we learn that. Protein denaturation, you learn protein denaturation. This natural conformation of the protein is lost, especially people due to the different denaturation agents. Let's see whether you remember the denaturation agents. What are denaturation agents that you can mention? Right? Denaturation is this loss of the conformation. Let's take that this one as the nice conformation, and then it loses its nice conformation. And then maybe those disulfide bonds are going to break, the ionic bonds and other weak interactions, they are going to break. So you say heavy metal, high temperature, organic solvents, strong acids, detergents, right? Detergents, they are the factors affecting on denaturation, right? Denaturation agents. You learn the difference between the denaturation and the digestion. Can someone tell me what is meant by digestion? So these are the amino acids present in the polypeptide chain. So in denaturation, there is no release of amino acids. No release of amino acids. Only thing is that the coiled special three-dimensional shape is lost. Special three-dimensional shape is lost. But if it releases amino acids, if it releases the amino acids, then you call that as the protein digestion. Then you call that as protein digestion. Protein digestion, right? Protein digestion. So you don't have to know the conditions for protein digestion. And do you know the condition for protein digestion? You can tell one condition because you have that condition inside your body. What actually helps in protein digestion? Give me one condition for protein digestion. Yeah? How protein digests inside your body? What actually helps? You learn that. What's the method? Huh? You learn it. What is it? No? No one knows. Poor. pH 2 of activate pepsin. Can you say it simple manner? Due to pepsin enzyme. That's correct. Due to pepsin enzyme. Enzyme, ne? Enzymes. You can say enzymes. You have pepsin enzyme, you have trypsin enzyme, chymotrypsin enzyme, peptidases are there. Enzymes can be right, helpful in digestion. There is another chemical condition also. If you heat with hydrochloric acid HCl at 120 degrees Celsius, you can do that. But anyway, that's not important because it's not mentioned in a resource book. But enzymes, you know that. Neither. Protein digestion. Protein digestion. Right, let's check your note. check your note. Strong acids digestion, okay ne? Digestion functions of proteins, catalytic protein, pepsin, amylase, catalyze biochemical reactions, structural proteins, keratin, prevent desiccation, collagen provides strength and support, and then storage, ovalbumin and casein, ovalbumin storage protein in egg, casein storage protein in milk, transport hemoglobin, transport oxygen and carbon dioxide, and serum albumin transport fatty acid. That's important, eh? Serum albumin transport fatty acid. Hormones, insulin regulate blood glucose level, glucagon also helps in maintaining blood glucose level. Contractile over the motor protein, actin, myosin, they are helping in your muscle function, and also people defensive myoglobin neutralizes foreign bodies. Okay, now people we come to the last discussion, nucleic acids. Nucleic acids. Nucleic acids. Acid inside the nucleus. Acid inside the nucleus. Now the classroom is okay, ne? Right? I can shift between the cameras and no trouble for you, ne? Everything is smooth now. I like to teach like this because I don't have much troubles. So improved compared to previous. So we can use this facility, ne? We can use this facility in future, right, to continuously support you to do the papers, because now we are going to do the paper number 2, ne? Paper number 2, and then you're going to answer it and we're going to discuss it. So once in 2 weeks time we have a paper, right? So we'll go for the real paper, right? Once you do the papers, couple of papers, we can go for a term test or something like that, ne? And then we can grade you, right? So so let's get ready, ne? Let's get ready. But most of the people are not here, ne? That's the worst thing. I think I have to take some action against those people who are not participating, right? Because they are going to find this is like too easy, and then they don't participate. But when we come into the advanced stage, because like this, when we continue at one particular point, people who participate, they will have good balance in their subject knowledge, and the others are going to be unbalanced, right? So then the things are going to change, right? So we have to get them. I'll take action to see that they are participating in this class, right? So bring your papers. You have to answer the paper. Bring the answered paper on Saturday to the class. I'll be checking them, right? If you don't bring the answered paper, going to be in trouble, little trouble, okay? So don't put yourself in trouble. Bring the paper, answered one. I'm going to check that. So you have now time on maybe on Friday you can answer, and then you can bring it on Saturday. We can discuss that on next Thursday. Right, people, nucleic acid. We learned that people, a special form of acid inside the nucleus. Acid of the nucleus. Because when they use basic dyes to stain the nucleus, so that the basic dyes stain the nucleus well. So they realize that some sort of acid inside. You call them as the nucleic acid. Now we know 2 types of nucleic acids. One, what do you call them? Deoxyribonucleic acid and ribonucleic acid, right? And then people as elements, we can see carbon, hydrogen, oxygen, nitrogen, phosphorus present as elements, right? And then people, we learn the unit. We learn people the basic unit which forms nucleic acid. What is the basic unit? What is the basic unit, right? You call that as a nucleotide, right, people? This is nucleotide. So you can see this structure of the nucleotide. Structure of the nucleotide, a phosphate group is there, sugar present, and you know this sugar is the ribose sugar, nitrogenous base present. How many nitrogen present in the nitrogenous base? How many nitrogen atoms present? 1, 2, 3, 4 10. How many? 5 neither. 5 nitrogen in nitrogenous base. This one is adenine, right? So you can see there adenine, but it's not fixed every time. If you take a purine and pyrimidine like that we learn. But anyway, you don't have to remember that now, but you need to know that there are a lot of nitrogen. So you call it as nitrogenous base. Nitrogenous base, right? Nucleotides without a phosphate is called as nucleoside. 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 Look at that. Nucleoside. Adenosine. Adenosine is a nucleoside. Adenosine is a nucleoside. Right? So you learn people phosphate. You learn phosphate. This is phosphate. This phosphate is reacting with a nucleoside. Phosphate react with the nucleoside. This is nucleoside CH2. This is the sugar part, and this is nitrogenous base. This is nitrogenous base. So this reaction forms an ester bond. This reaction forms an ester bond, right? The reaction forms an ester bond, right? So there is an ester bond which form here. Right? Let me show that ester bond. So the nucleoside, as it become a nucleotide, an ester bond is going to form. An ester bond forms, right? I forgot to mark here. This is OH, hydrogen. It can be OH if it is ribose. If it is ribose, it looks like that. So this is the nucleotide. This nucleotide, 2 nucleotides can react. 2 nucleotides can react. And then you learn people that there are several different types of nucleotides present. How many different types of nucleotides present in a cell? Can you answer my question? How many different types of no. Nucleotides. Nucleotides I'm asking. How many different types of nucleotides present? Only one phosphate type, phosphate only one. Then sugar types 2. There are 2 sugar types, ribose. What is the other one? Deoxyribose, 2 sugar types. Deoxyribose, ribose and deoxyribose. This is deoxyribose. Then how many different nitrogen bases, my dear? Nitrogen bases: adenine, guanine, cytosine, thymine, uracil, 5. So then you learn phosphate with ribose and that with adenine. Phosphate, ribose, adenine. Phosphate, ribose, guanine. Phosphate, ribose, cytosine. Phosphate, ribose, uracil, Phosphate, ribose, uracil. Then phosphate, deoxyribose, adenine. Phosphate, deoxyribose, guanine. Phosphate, deoxyribose, cytosine. Phosphate, deoxyribose, thymine. Phosphate, deoxyribose don't go with uracil. Same like that, ribose don't go with thymine. So altogether how many nucleotides then, my dear? 8 different types of nucleotides present. 8 different types of nucleotides present, right? 8 different types. So you learn the names of those 8. You learn the names of those 8. Remember the names of them. So let me show that. Let me show it. Let me show them. The nucleotides in RNA, nucleotides in DNA. adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, uridine monophosphate. Ne? What do you have here? Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. You don't have to remember, but I think you better know the names. You better know the names. So 8 different nucleotides are there. Please check your note. We wrote the full term, right? We wrote the full term, right? Nucleic acids. And then people, when nucleic acids form polynucleotide chain, nucleic acids form polynucleotide chains. Formation of nucleic acid. Formation of nucleic acid. Now check that part. Check that part. Pentose sugar, nitrogenous base. Purines and pyrimidines are there. Nitrogenous bases, purines and pyrimidines. Forgot to mention that. What are purines? Adenine and guanine are purines. Pyrimidines, what are they? Cytosine, thymine and uracil. And then phosphate group. Phosphate group. Formation of nucleic acids. Right? Millions of nucleotides. Millions join. Now we don't see that for proteins. Students, now we don't say that millions of millions of amino acids are joined to form proteins. We don't say that because it's 100 to 300, not more than that. So we can't say that millions of amino acids join with one another to form proteins. That is wrong. You can say millions of nucleotides are joining to form nucleic acids. That's correct. Now in DNA people, we can see roughly around 8 million nucleotides present, join one after the other, right, to form a DNA molecule. 8 million, so that's like millions. So it's correct here. Millions of nucleotides join by phosphodiester bonds to form polynucleotide chains. Condensation between the OH groups of the phosphate of one nucleotide and OH attached to the third carboxyl of the pentose sugar of the other. Okay, let's see how that happens. Let's see how that happens. Right? Let's see how that happens. When 2 nucleotides are joining, right, the special event happen. 2 nucleotides are going to join. This is phosphate, this is ribose sugar, this is the nitrogenous base. I'm going to take NB. This is another phosphate, this is another sugar, this is another nitrogenous base NB. So what happened now? They say that OH group of the phosphate, they say that this OH of the phosphate, OH of the phosphate of one nucleotide and OH of the third carbon atom. This is the third carbon atom OH. You learned people the numbering of carbon, 1, 2, 3, 4 and 5. That is how you number it. So third OH and the phosphate OH, they react. They react and then forms a new bond. This bond is also ester bond. This is also ester bond. You learn already there is an ester bond here. There is an ester bond here. So this sugar and this sugar both are connected by 2 ester bonds with a phosphate. What do you call? Phosphodiester bond. Phosphodiester bond. Why? 2 esters and 1 phosphate present in between. This one is also ester. Ester bond. So 2 ester with a phosphate at the middle. Ne? So this reaction can repeat because you know this point has another OH, that has another OH. So when another phosphate come and interact below, when another nucleotide come and interact below, the reaction can repeat. Reaction can repeat, right? The reaction can repeat. Now look at this. Reaction can repeat again and again and again and again and again and again, millions of times. It can repeat, forming the polypeptide chain. Yeah, forming the polypeptide chain. So we get the polypeptide. Ne? So we get this polypeptide, right? This is the polypeptide. So we obtain this polypeptide chain. This is the polypeptide, ne? Phosphate and sugar, nitrogenous base, phosphate and sugar and nitrogenous base, phosphate and sugar and nitrogenous base, phosphate and sugar and nitrogenous base, right? So this can repeat, forming a long chain like this. This forms a long chain like this, phosphate and sugar, right? It forms a long chain like this, right? Connected with what? These bonds, connected with bonds. So this is a polynucleotide chain, ne? These nitrogen bases can be adenine, guanine, cytosine, adenine, guanine, cytosine. So you get a polynucleotide chain. What do you call this part, people? Can someone tell me what this is? What this is? Come on, type that. What is that part? What do you call it? What do you call it? It's a backbone. Ne? What backbone? Now you learn backbone earlier also. Ne? You learn earlier backbone for protein. Now what do you call this one? Sugar phosphate. Ne? Sugar phosphate backbone. Sugar phosphate backbone. This one is sugar phosphate backbone, right? This is sugar phosphate backbone. This is sugar phosphate backbone. Sugar phosphate backbone, right? And then this is the RNA structure. Ne? If you take a DNA, what do you see, people? Two of them join together. Ne? Two chains like this interact with one another and then forms the DNA molecule. Then forms the DNA. Ne? Then forms DNA. Right? Two chains are going to join like this. They are opposite in direction. They are opposite in direction. Right? The directions are different. And phosphate. Ne? These are phosphates. Two chains join with one another and then forms, people, the DNA structure. Ne? Watson and Crick describe this model. Every time when adenine present, thymine on the other side. Every time when guanine present, cytosine. Cytosine present, guanine. Adenine present, thymine. Guanine present, cytosine. Cytosine present, guanine. And we learned the number of double bonds also. Ne? Number of hydrogen bonds, adenine, thymine 2, guanine, cytosine 3. Guanine, cytosine 3, adenine, thymine 2, guanine, cytosine 3, guanine, cytosine 3. This is another backbone. This is another backbone. We learned that this end has 5-N. This end is ending with the third carbon atom. So this is 3-N. How do you pronounce it? 5' and the 3' ends. This one is on the opposite direction. This side is 3', this side is 5'. Ne? This side is 3' and 5'. So we say that these two strands are opposite in direction, right? And matching. What do you call matching, people? What is the term that you used to say matching? Right? They are complementary. Right? Complementary. So you learn the DNA structure of the DNA molecule. Structure of DNA molecule. Right? Structure of the DNA molecule, Watson and Crick model. Watson and Crick model. So look at this. Structure of the DNA molecule, Watson and Crick model. Watson and Crick model. DNA molecule have two antiparallel polynucleotide chains that spiral around an imaginary axis. Spiral because the DNA molecules twist like this, ne? They twist like this. Two strands are not straight, twist like that. So you call spiral, ne? This is 3' and the other one has to be 5'. Ne? Two strands spiral around each other, and therefore this forms people the double helix structure. The two sugar phosphate backbones run in opposite direction from each other. So this is sugar phosphate backbone, sugar phosphate backbone. So two of them present. This is also sugar phosphate backbone. Adenine pair with thymine, guanine pair with cytosine with 3 hydrogen bonds like that. The sugar phosphate backbones are on the outside and the nitrogen bases are paired in the interior of the helix. Two strands are held together by hydrogen bonds. Base pair rule, look at the rule people. Thymine, adenine, 2 hydrogen bonds, guanine, cytosine, 3 hydrogen bonds. Then hence the structure of the DNA are said to be complementary to each other. These pairs are known as complementary base pairs. In this original double helical structure, one complete turn consists of 10 base pairs shown in the diagram. So this is the DNA double helix. This is DNA double helix, DNA double helical structure. Right? Functions of DNA: store and transmit genetic information from one generation to the next. Store genetic information for protein synthesis. Structure of the RNA structure people you learn, it is a single strand. It is a single strand, 5' and 3', single strand like this, right? And it can have different arrangements. It can be straight, it can be sometimes coiled like this. Now, for example, tRNA, it coils like this. tRNA, it coils like this. So, some regions are double stranded, some regions are double stranded, right? Now, these parts, I'll color, these parts are double stranded. These regions are double stranded, right? These regions are double stranded. So, some double stranded regions are there. So, there are 3 types. What are those? Messenger RNA, transfer RNA and ribosomal RNA. So, look at them. Messenger RNA is a linear molecule. Linear means unbranched, and there are functions: copy genetic information stored in the DNA, transfer genetic information. Transfer RNA, tRNA, look at these people. It has a loop structure, the 3 loops are there. So, it transport amino acid. You know, amino acid go and bind here, right? So, its job is to transport amino acid during protein synthesis. You learn protein synthesis brief, and then ribosomal RNA. They are structural component of the ribosome, right? Differences between DNA, RNA. DNA is double stranded molecule. RNA consists of adenine, thymine, guanine, cytosine and absence of uracil. Sugar molecules in RNA is ribose. DNA has deoxyribose. You know, nitrogen bases, thymine, uracil, difference is there. Thymine, uracil, difference is there, right? So, that is all for the DNA structure. One more part, that is nucleotide. That is interesting. Check that part a little bit, right? This is useful. Nucleotides other than those found in nucleic acids, ATP, NAD plus, NADP plus, FAD, function, universal energy carrier. NAD, you understand this better, ne, the people? Now you understand it better, ne? Now we were discussing about electron transporting, ne, the electron carriers, oxidizing agents. Now you understand with the photosynthesis lesson. Now you have a good feeling, ne, when we say electron transport electrons in the between reactions, ne? NAD becomes NADH and then transport electrons from the light dependent reaction to the Kelvin cycle, ne? Right. So, it is a electron carrier, and then also people, it helps in the reduction steps in the Kelvin cycle. Reduction step in the Kelvin cycle, NADPH. So, it is an oxidizing agent. It is an oxidizing agent. NADP plus act as an oxidizing agent and a reducing agent. Ne? So, NADH is a reducing agent. NADP plus is an oxidizing agent, right. So, you learn them here in this lesson, but now we learn about their true involvement people in the photosynthesis lesson. Some more about NAD we are going to learn people in the coming Saturday with the cellular respiration lesson we are going to learn, and then we are going to meet again people on Monday for the couple of extra classes also, right. So, let your friends know that we are starting unit 3 in the extra session, right. Extra class will be starting unit 3. So, let your friends know that. For the new students, they can come and join with the class, right. So, it is a good point to start unit 3, right. Okay. So, functions of FAD act as coenzyme electron carriers. You will understand these things better when I discuss the cellular respiration lesson. Okay. So, then people, that is all for the day, right. That is all for the day. Thank you very much for staying with us with patience, and learning, revising this part. Give me your suggestions and your comments on this work, right, whether it is useful or not. Let me know, right, comments how we should improve it further, right, for you. Let me know. So, we will meet then on Saturday, right. Thank you very much, and then I will see you soon. I will see you soon, right. Okay then, bye bye. Okay then, see you, bye.